Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today we'll discuss a very important topic and a very important concept with you people. And the concept is that why we use 70% alcohol instead of 100% alcohol. Means that why we use diluted alcohol instead of concentrated alcohol to disinfect a surface or something. Okay. Basically, what is disinfection? I want to tell you that if the you are you often clean your kitchen surfaces, or when you are working in the laboratory in the laminar flow hood. So first you spray alcohol over it so that the microorganism that are present inside it should be killed by that chemical. Okay, that spray. So it is basically known as disinfection. Just look to these images. Okay, this is a type of spray that can kill the microorganism. Here is also a type of spray. So basically we use alcohol to disinfect surfaces. Okay, so basically we use diluted alcohol to disinfect the surfaces instead of 100% alcohol. So there is a question that why we use 70% alcohol rather than 100% alcohol. Question arises that most of the student it comes in their mind that alcohol, if something is concentrated, so it can act well in that activity, but instead of concentrated we are using diluted. Okay, so here in this video, I will explain the basic concept that why we are using 70% alcohol. So after watching this video, they will come to know that why we are using diluted alcohol. I will explain this concept in my way. So stay tuned with the video and watch the video till the end. Okay, don't look to the duration of the video that because I am explaining it in a detailed way, so so that you could answer this question to your examiner or to someone else. Okay, if this question is asked from you people in the exams or from by someone else. First of all, you should know about the disinfection. As I told you, that disinfection is the destruction or removal of all pathogenic organisms or organism capable of giving rise to infection, but not necessary spores. Okay, keep this thing in mind when you are disinfecting a surface. Okay, so your aim is to destroy all the pathogenic microorganism. Suppose you are working in the laboratory and you are performing an experiment. So first you disinfect that surface so that the microorganism that are present on that surface should not interact with your experiment. Okay, but keep this thing in mind, disinfection does not kill spores. Okay, with the process of disinfection you cannot kill spores because spores are very much resistant, and it can be killed by the autoclave, by the sterilization process. Okay, but this infection is quite useful and it can work well. Basically disinfection is the destruction of or removal of all pathogenic organism. Okay, from a specific surface. Okay, I want to clear to you people that how alcohol acts on protein. Okay, I add this slide because this will help you in the later video. Okay, just I want to give you concept of this, that when you add alcohol to a specific protein. Okay, for example, there is a mixture of protein and you add alcohol to it, either it is concentrated or diluted. Okay, but you add alcohol to it, for example it is concentrated. So what is the action of alcohol to proteins? Okay, what is the action that alcohol perform on protein? So basically when there is concentrated alcohol or it is for example diluted, so how it acts on the protein, it basically denature the protein. It for example, this is a compatible protein, and after when alcohol react with it, so it denatures the protein, it damages its function, okay, it damages its structure, it if there are many proteins, so it coagulate that protein, in simple that it makes it we can say a malfunction, or we can say that it destroy the protein in different ways. But you should kept this in mind, when the higher the alcohol, the concentrated alcohol, the more easily will it denature the protein. So if you add 100% alcohol, a highly concentrated so it can denature the protein within seconds. Or if you add diluted alcohol, okay, so it can take some time to denature the protein. So first of all, keep this concept in mind, that when you add alcohol to a protein mixture, so it denatures the protein, it destroy the structure of the protein and making them unfunctional, okay. So I was just telling you about this. It will help you in the later video. Okay, first of all, I want to clear the peptidoglycan layer. As I have told you that I have made this lecture in my way, so just stay tuned with this video. In the last, you will get your answer. But first of all, I want to make a background for you people. So first of all, we will check on the peptidoglycan layer. I am discussing peptidoglycan layer because mostly there are bacteria, pathogenic bacteria, gram positive, gram negative bacteria that is killed by the process of disinfection. So if you keep this thing in the structure of peptidoglycan in your mind, so then you can easily understand the concept. Okay, basically this is the molecular structure of the peptidoglycan layer. Okay, this is the molecular structure, and this is the main structure. I just add the slide because I want to show you the protein content in the peptidoglycan layer, the amino acid content in the peptidoglycan layer. Just look here, it is a simple chain of peptidoglycan, a single chain. So here is N-acetyl glucosamine and N-acetyl uramic acid, although it is not amino acid. After that, this is also an amino acid, this is also amino acid, and this is also amino acid. And this chain is interconnected with another chain and with the help of 5-glycine amino acid. Okay. So I was just showing you the protein content, the amino acid content in a single chain or a double chain of a peptidoglycan, that how much high content of amino acids are there. And basically, what are amino acids? When they combine, they form protein. So these are basically the amino acids. These are the amino acids. Okay. So I was just showing you that in a peptidoglycan structure, there is a high amount of amino acid. Keep this thing in mind. Okay. After that, this is the gram positive bacteria, the cell wall of gram positive bacteria and the cell wall of gram negative bacteria. Now I will show you that first of all, we take concentrated alcohol. Okay, for example, if we take 100% alcohol, okay, it is not diluted with the water, and we take 100% alcohol. So why now here the question that I have asked in the beginning, it will be clear to you. If we add 100% alcohol and want to disinfect a surface, so what will happen when we add alcohol? So it will start if there are gram positive bacteria. Okay, if there are gram positive and gram negative bacteria on the surface, so how will concentrated alcohol work? What will it do if it enters the peptidoglycan layer? So just because of the high concentration of alcohol, it will directly within seconds start coagulating the proteins. Okay, that are the peptidoglycan layer basically. Okay, so it will directly start denaturing the proteins, coagulating the proteins. Okay, it starts coagulating and denaturing the proteins. So before entering and getting access to here, okay, before getting access to here, what will happen? it will just if it goes inside just like this and start diffusion. Okay, so within in the middle of the peptidoglycan layer, due to the high concentration of alcohol, it will directly coagulate the protein. And when it coagulates the protein, so basically when the protein is denatured and coagulated, so the spaces that is in between these peptidoglycan layers, they are acquired by the denatured protein. Okay, first of all, for example, this protein is in this shape. After when it is coagulated, so it change its shape. And if I want to change the color, so it's changed its shape, and it just look to the yellow color to my pen. Okay, it when it is denatured directly, so it just block this pathway. Okay, is just because it has changed its structure. So the free spaces that are present inside these peptidoglycan layer are acquired by the peptidoglycan, by the structure of the by these peptidoglycan. Okay, oh, and basically what are these are the amino acids, the protein the components of protein. So these are denatured. Okay, so they acquired a different position in the peptidoglycan layer just because their structure has been denatured. So these spaces are filled by the denatured protein. So this alcohol cannot further get access to the cell. Okay, it should be just blocked here, or for example here. Okay, if it get access to the periplasmic space, so here in the plasma membrane there is also a very high content of protein, the integral protein, the extrinsic protein, intrinsic protein. So they are also directly coagulated by the concentrated alcohol. So what happens if it get access through the periplasm to the from the peptidoglycan layer, so it can be blocked here by the other proteins. Okay, because they directly denature this protein, so it cannot enter the cell. It cannot enter the cell and cannot destroy the DNA and the cellular organelles of the cell. So just because of this, the cell can survive because its DNA content is already we can say that it is the DNA content is not destroyed by the alcohol. Even the organelles are also not destroyed by the alcohol, because this alcohol has not get access to inside the cell. Because the main reason is that due to its high concentration, the concentrated form, it is blocked in the middle of the peptidoglycan layer or the plasma membrane, because there is a high content of protein, and these protein are directly within seconds coagulated by the alcohol. So they block the pathway and it prevents the further diffusion of the alcohol. So just because of this, the cell survives. Okay, the cell survives, and after that there is a mechanism that it can then again repair its peptidoglycan layer in the proteins. Okay, so the microorganisms survive. Here is also the case or in the gram negative bacteria. When we add 100% alcohol, so these are the proteins, the porine protein, the lipopolysaccharides, the peptidoglycan layer, the integral proteins, and the phospholipids. So these all are directly coagulated by the 100% alcohol. So they cannot get the alcohol cannot get access to the internal cytoplasm, and the DNA cannot be destroyed, and the organelles can also be cannot be destroyed, and the cell can survive. Okay, so this is basically we do not use 100% alcohol. For example, if we are using 70% alcohol, so what happens, as I have told you that if it is diluted, so it coagulates or it denatures the proteins in its pathway, but it is very they coagulates the protein very slowly because it is diluted. So slowly they coagulate the protein, but it gets an ample time to reach the cytoplasm. Okay, because if it is has 70% alcohol, so if it is entering and start diffusing inside the cell and the peptidoglycan layer, So rather, for example, if we add 100% alcohol, okay, so it is coagulating within seconds the protein. But when we add 70% alcohol, so it is coagulating the proteins, the denaturing the protein, but it is taking very much time. Okay, so because of this taking time, it can get access to the inside of the cell, and after that it can cross the plasma membrane, and it can enter the cytoplasm and destroy the DNA and cellular components, okay, the organelles, okay. So just because of this, we use 70% alcohol instead of 100% alcohol. Hope you have understand the idea. It is of for the gram positive and gram negative bacteria. If we take fungal cell wall, so the same is the case with the fungal cell wall. These manoproteins are the proteinaceous components that are present on the surface of the cell wall of the fungal fungi. So if we add 100% alcohol, so these proteins can be directly coagulated, okay, directly coagulated. So it cannot enter the inside the cell, okay, if I want to show the cross. And if we add 70% alcohol, so what will happen? It will slowly even denature the protein, but it can take some time to enter the cell, okay, and destroy the cellular components and the DNA and the genome of the fungi, okay. So this is also for the fungi. If we take viruses, so viruses can also be killed by the alcohol, but it should be 70%, because look to the surface of the viruses. These are, and it is having high content of protein. If we look here, HIV virus, so these spike proteins and these glycoproteins, okay, these are proteinaceous compound. If we look to influenza, the hemagglutinin, the neuraminidase, okay, these are proteins in nature. So if we add 100% alcohol, so directly this protein will be coagulated, and it will protect the internal environment, and it will prevent the further entry of the alcohol, okay. And if we add 70% alcohol, so it will take some time, and it will enter the inside the cytoplasm of the cell, the virus, and it can destroy the genome of the virus, okay. So I have explained it in my way. If you have any question, you can ask me in the comment box. I hope you have understand the main idea, that why we use 70% alcohol instead of 100% alcohol. So if someone asks from you, so you can directly answer their question. And if he needs an explanation, so just explain it in your way, okay. Because science is a type of a subject that it can be explained, anyone can explain. If you understand it well, so you can explain it well, okay. So hope you have get the concept, okay. So thank you very much, and share this video, like this video, and also comment on this video, and subscribe to my channel to get more interesting videos and interesting concepts related to science. Thank you very much. God bless you.